Hi everyone. So now we've reached time for your first lecture exam. So I want to say a couple of things. This video is going to be a longer video because I'm going to go ahead and try to sum up or recap all of the first 4 lessons. But I would strongly encourage you do this in short chunks, right? Do it maybe 10 or 15 minutes, maybe 20 minutes, and then come back and do some more later. Your brain is only going to absorb so much of this at one time. Also, while you're watching this, when I say pause and try to recall or do it on your own, please take advantage of that. That's your opportunity to sort of have a self-check, see what you know, start practicing that recall information. Also, if you can sketch, draw, write, that is going to help make connections also. So instead of just typing on your computer, studies have shown actually physically writing or sketching is going to be a great way to remember the information. So let's get started. So the scientific method, so again, you have a study guide to follow along. You've had all the content in the lessons. I'm going to try to hit the high points and just know that there may be a few little things that we don't cover in this review, but we're going to try to hit the most important things. So scientific method, right? It's a process we follow in science. We know that step 1, we have to come up with what? What's the first thing that's going to be the first part of the scientific method? Yeah, right. We maybe have a problem to solve or we observe something. And so then we're going to create a hypothesis, right? An if-then statement. This has to be something that's testable, right? That's clear. We have to be able to disprove this hypothesis. Now, to do that, right, we probably are going to design an experiment, and this experiment's going to contain variables. So if, in fact, I'm interested in growing tomatoes, okay, and my observation or my question is, is one of these plant foods better? So let's say I design my experiment. So my hypothesis is, if I use Miracle-Gro, I will have a better tomato crop or a better tomato yield. So I'd have to define what that means, right? Healthy tomatoes, more tomatoes, whatever. What would be my independent variable in that experiment? So my independent variable is going to be the plant food, right? So am I going to use Miracle-Gro or another brand? Or am I not using it, right? Am I using manure? The kind of plant food would be the independent variable. My dependent variable is going to be, it depends on the independent, right? This is what I'm measuring. So if I decided on the number of tomatoes or how healthy they are, how juicy, right? Those are my dependent variable. What about standard or controlled variables? What would be some examples of those? So there's a number, right? Some of the things would be the amount of sunlight, the amount of water, right? The basic kind of soil, the type of seeds. Did I start from seeds? Did I start from a prepared plant? Those would all be things, right, that would be our standard variable. So make sure that you can identify those different types of variables from different situations and conditions. And just keep in mind the independent one is essentially the one that you're testing, right? That's the one you're manipulating or changing as the experimenter. All right, so as part of an experiment, you're going to be collecting data, right? So in the last thing when we talked about our tomatoes, our data might be how many tomatoes were produced over a given time, or the size or the weight of those tomatoes, right? We would want to collect some kind of data. Now, when we report on the data, sometimes the data will be graphed. So in other words, we'll put it on some kind of a graph. And typically your independent variable, which I just want to make sure I reiterate, you should have one independent variable, right? If we were testing the amount of water and the amount of sunlight and the type of plant food, how would we have any idea at the end which one of those was making the difference, right? So one independent variable at a time. That will go on the x-axis, right? The horizontal axis in a graph. And our dependent variable then goes on the y-axis or on the vertical axis. Okay, we talked through some standard or controlled variables, but I also want to make a note about what is a negative or a positive control. So that's different than a controlled variable, right, or a standard variable. So we would have a positive control and we would have a negative control. So as we go through this class, you're going to see that you'll have experiments that we talk about those kinds of controls. So A negative control means I'm not exposing it to my treatment, right? So if in the Miracle Grow tomato, maybe I would have one plant that did not get any plant food, right? It had soil, it had water, it had sunlight, no plant food. There's my negative control. My positive control would be something that you expose to something that is a known normal level. So that one's a little bit harder because we were testing different brands, okay? But I might have, maybe I have a current plant food that is commonly used. That's my normal, and I'm testing some other brands to see if they perform better. All right, when we looked at lesson 1, we also talked about the characteristics of life, right? There were several characteristics of life. So what I want you to do right now is just pause the video for a second, get a little piece of scratch paper, see how many characteristics of life you can jot down. Now, what do I mean by characteristics of life? I mean, these are the things that are common for all living things, right? So for example, movement or motility, that's not a characteristic of life because not all living things move, right? But we had several. So see if you can jot those down. Once you do, unpause the video. So one of the characteristics of life, right, is having order or organization. And so we're going to talk through the levels of organization in living things. And right here, I have some of these filled in already for us. So atoms, right, that's sort of the smallest particle that we can get to that has the same properties. So we have lots of different elements that are made up of atoms. So we have hydrogen atoms, we have oxygen atoms, we have carbon atoms, right? We talked, and we'll talk some more about that later in this review. Atoms then can bond together to form molecules, right? And we can have molecules come together to make much larger macromolecules, organelles, larger things, and eventually, right, we have these molecules organized into what we call cells. Now I'm going to put a little asterisk here by cells because this is the smallest unit of life, meaning nothing smaller than a cell do we consider living. Now, if we have multiple cells working together or coming together to form the next level of organization, what would that be? Yeah, that's tissues. Okay? Then if we have multiple tissue types coming together to form the next level that has abilities and characteristics, right, that can carry out functions that tissues alone can't carry out, what would that be? So that would be organs. And then we have multiple organs coming together to form an organ system, right? Then we have multiple organ systems coming together that form an organism. Now I want to stop here for just a second because I want to make a note, and we're going to talk about this in the next slide. But some full living organisms, right, this is it. This is all they get to. They get to the cell. Pardon me. They don't have tissues. They don't have organs. They don't have organ systems, right? Some organisms are only single-celled. But complex organisms can have tissues, organs, organ systems, organisms. Now I'm going to draw a line here, okay? This line is the difference between an individual living organism and groups of organisms. So this next category would be when you have multiple living organisms of the same species in the same place at the same time that can interbreed. So this is a population, right? So if I were to walk outside in my yard, right, I would have multiple populations. So there's a population of grass. There's a population of many different populations of birds, right? Sometimes I see cardinals, sometimes just regular sparrows. There are populations, I see some frogs sometimes, obviously different bacterial species, lots of different populations. Now, the next group of organization would be all of those populations coming together. So all the living things in my yard or in my neighborhood area would be the community, right? All living things. The next level up from community would include things that are not living: air, water, soil, rocks, and that would be the whole ecosystem. And if we look at all the ecosystems on the surface of the earth where there's life collectively, then that would make up the biosphere. Now I want to spend a little time camping on cells as it relates to organization. Now, as we go through this review, when we get to lesson 4, we're going to talk even more about cells and organelles. But when we're thinking about organization, one thing we need to keep in mind is we have 2 basic cell types, right? So take a minute, pause this, jot down in your notes, what are the 2 basic cell types? Now, I'm not talking about animal and plant cells. They both belong to one of these basic cell types. So make a pause and let's come back to this. So they are prokaryotic and eukaryotic cells, right? What is the biggest difference between these 2? So eukaryotic cells have membrane-bound organelles. In other words, there are these small little membrane-bound components of these cells that carry out specific functions. The prokaryotic cells don't have those. Now, in addition, eukaryotic cells, are they larger? Yes, they're typically larger. They're typically more complex because think about all the organelles, right? And eukaryotic cells form multicellular organisms. Now, we can have single-celled eukaryotic organisms, but the majority of them form multicellular organisms, whereas prokaryotic cells are going to form our single-celled organisms. Now, these are the 2 cell types. But as we're talking about organization, right, there are 3 domains that all living things are categorized in. So if we look at all the living things and we start plopping them into these categories, they all go into one of these 3 domains. So what are they? Take a minute, jot it down. All right, so we have archaea, we have bacteria, and we have eukarya. So eukarya sounds a lot like eukaryotic, and guess what? Yep, it's made up of organisms made of eukaryotic cells. Archaea and bacteria both are made of prokaryotic organisms. So all living things are going to fall into one of those 3 domains. All right, now pause the video, look at all of these pictures, and see if you can come up with 1 or 2 characteristics of life that you could apply to the particular picture that you're looking at. Okay, and then come back and we'll talk them through. All right, in this first one, we see a little baby elephant, right, along with probably mom. So which characteristic of life? We could think about a couple, right? We could think about growth and development. That's one characteristic of life. And certainly that baby elephant is undergoing that. We could think about reproduction, right? Because obviously this little baby elephant went through the reproductive process, right? So started out as a zygote, a single cell, and that cell divided and it developed, and here we have a baby elephant. All right. What about this picture here that has all the food, and it's pretty close to lunchtime, so I would really like to have one of those hamburgers right now. All right. So this could be, right, energy processing, because we need to take in calories and nutrients, right? So our cells have that to produce the energy in the form of ATP that we need. We also need nutrients for vitamins for our enzymes to work correctly. So it could be energy processing. Okay. Let's see. What about this picture here with sort of the moth/butterfly? Okay, so this one might be a little trickier. So this one could be considered adaptation, right? Or evolution, because this little coloration looks like the eye of an owl. Well, the predators of these butterflies or moths, right, are birds. They eat them. But the predators of birds, right, are owls. So when this eye spot, when the wings move quickly and that eye spot is flashed, it might appear to a predator that that's an owl, and so they might actually leave. So that might be an adaptation for survivability, right, of these against their predators. All right, what about this one with the sweat? Okay, so this one could be homeostasis, right? Because we have to maintain homeostasis in a lot of our bodily functions, including our body temperature, and sweating is one of the ways we do that. You could also think of it as energy processing because clearly we're having to use our muscles, right, as we're working out, which requires energy. So our cells must be functioning in order to produce that ATP energy that we need. And so this is a model of a heart. This one I would think of as meaning order and organization because it represents an organ, right? That's part of an important organ system for our body. So now we're moving toward lesson 2, and you see here the familiar periodic table with all of the elements listed. There's no memorization that needs to happen for all the elements or anything like that, but I do want you to be able to understand some things from the periodic table, and let's talk it through. So we've chosen hydrogen, right, which is the first element in the periodic table, and we've sort of blown it up for you so we can look at what's included in each of these atoms in their little box of the periodic table. So atomic number, that's important. We need to understand what the atomic number means. So first thing, as we see, it goes in order, right? Number 1, number 2, number 3 on the next row. So we know that elements are in order based on their atomic number. What exactly is the atomic number? Okay, the atomic number is the number of protons. Now, pause your video, jot down what do you know about protons, and then come back. All right, so protons are the positively charged subatomic particle, right? We have a couple. We have protons, we have electrons, and we have neutrons. So the atomic number tells us the number of protons. So that means all atoms of hydrogen have 1 proton, right? If it has 2 protons, it's not hydrogen, it's helium, okay? Now, obviously we have the symbol and we have the name. The other number is called the atomic mass, okay? So is a different number, means a different thing than the atomic number. The atomic mass is literally the mass of the atom, right? And the mass is equal to the protons plus the neutrons. Not the electrons, right? The protons plus the neutrons. So in this case, we have 1 proton and no neutrons. So we usually round that number, right, when we're trying to figure out how many protons and neutrons. So to pick another one, I'm going to use carbon. I'm going to circle carbon right here. And I know it's small, so I'll tell you what it says. So it says that carbon has an atomic number of 6 and has an atomic mass of 12. Now it says 12.01, but I'm going to round it to 12. So that tells me the mass of carbon is 12, right? And I have 6 protons. So how many neutrons must I have? Well, if I have the mass of 12 minus 6 protons, I have to have 6 neutrons. Now, carbon, this 12.01 is an average of all the carbon atoms out there. So that means some of them are going to have a mass of 13. Some are going to have a mass of 14. And that means that they have a different number of neutrons. They have to have 6 protons, right? We know that. If it had 7 protons, it would be nitrogen. If it had 5 protons, it would be boron. So it has to have a different number of neutrons, okay? We're going to talk about that in the next slide, but having a different number of neutrons means it's an isotope. That doesn't mean there's one normal form and then there's all these other, every form of carbon. So carbon with 12, mass of 12 is an isotope of carbon. Carbon 13, that means a mass of 13 is an isotope of carbon. Carbon 14 is an isotope of carbon. It's a different form, and those forms are based on the fact that there's a different number of neutrons. All right, let's move forward. This one's going to be probably the longest pause I'm going to ask you to take. And I know you're probably going to think, I'm just going to move forward. I don't want to do this. But it really will help you to make connections, see what you know. I don't know if you've ever used a concept map. I love concept maps. I'm a terrible artist, but I like to do things visually. So a concept map works great for me because I don't have to draw anything pretty. I can just make connections. All of these terms are related to atoms and bonding and the chemistry that we have to learn in this class. So make yourself a concept map, meaning make connections, make arrows, start making sense of this. I'll start it for you. You can do yours in a completely different way, okay? But I don't have much room here, but let's say that I put atoms right here in the middle, okay? And then I'm going to make a little bubble off of that, and I'm going to call it subatomic particles. I don't even have that listed, but you can add to it, okay? Now I'm going to do 3 little circles off of that because I know that electrons are 1 subatomic particle, protons are another one, and neutrons are another, okay? Now, I'm going to write on this line right here that goes out from subatomic particles something that I want to know, which I don't have much room, but I'm just going to put orbit, right? Because I know the electrons are not in the middle of that atom. I know they're in constant motion orbiting. So that's just a little reminder to me, okay? Now, I'm going to also make a note that electrons have a negative charge, protons have a positive charge, right? And neutral, I'm just going to put no charge. They don't carry a charge, okay? Now, the other thing I'm going to do, I'm going to come off of electrons, right? And I'm going to make I'm just going to make this a rectangle, and I'm going to say bonding. And why did I say that? Because electrons are what are either shared or transferred. Whoops. That's what makes a chemical bond. Bonding occurs, right, between electrons. Then I'm going to come off of bonding, and I'm going to have a line for covalent, and I'm going to have a line for ionic, and I'm going to describe what those are. That's a concept map, right? Putting these things together. So pause it, come back, and then we'll finish out the chart together. Okay, welcome back. I hope you did your concept map. Covalent bonds occur when atoms share electrons, right? Ionic bonds occur when atoms transfer electrons. Now, my little picture is in the way here, but what I need to do with ionic, I need to make another little circle, and I need to say ions, right? Ions are what are formed whenever an ionic, or ions are what forms an ionic bond, right? When atoms transfer electrons, you're going to end up with a positively charged ion, which is a cation, and a negatively charged ion, which is an anion, and the attraction of those two, right, that's what forms the ionic bond, okay? Now, right here off of bonding, I'm going to put octet rule. And if you go on to take chemistry, you'll get into much more detail about electrons and why atoms share and why they transfer. But the octet rule means atoms want to have a full valence shell, okay? Valence shell is just the outer shell where those electrons are orbiting, right? And so octet comes from 8 because many of those shells hold 8, but we also know the first shell is full with 2, and then some shells actually hold more than 8. So octet, even though it has the word 8 in it, it means the valence shell is full. Okay, now I have a couple terms down here that I haven't used, right? Mass number or atomic mass and atomic number. And I also haven't used periodic table, right? So I'm going to have to work those in some way. So what I might do is I might say periodic table of elements. And then I would say, okay, it's an atom is the smallest piece of each of those elements, right? And from the periodic table is where I'm going to find my atomic number, which we know is protons, okay? And my atomic mass, which is protons plus neutrons. Okay, now let me see, have I covered everything? No, I have not. I have not covered isotopes. So now we know from the previous slide, it's neutrons, right, which form isotopes. So I would put a note here, isotopes off of neutrons, and I would probably make a little note off to the side and say isotopes occur because there's different numbers of neutrons, right? So an example might be carbon 12, carbon 13, and carbon 14. Okay, so this slide brings us to the atom and the subatomic particles of the atom. Now, the takeaway things, right? Let's talk about what particles are inside the nucleus. Now, the nucleus of the atom is not the same thing, right, as the nucleus of a cell. In this case, nucleus just means the core, right, the center. So we have this one particle that doesn't have any charge written on it, right? Those are the neutrons. We have these particles with the plus signs written on, those are the protons. So those are all in the core of the atom, and these are the ones, right, that we would add together to get the atomic mass of the atom. And then we have electrons, okay, negatively charged particles, and those are shown in this orbit, right, of the atom, and because they're in constant motion, and we think about those in terms of energy shells, right? So this would be that first energy shell, and it's got 2 electrons in it. Now, the protons, right, that are in this nucleus, those are the ones that are the atomic number, okay? So the number just contains the number of protons, whereas the mass is the protons plus the neutrons. So an ionic bond, we talked about 2 different types of bonds, right? We have covalent bonds and we have ionic bonds. Ionic bonds are when electrons, which sometimes I'll just do E minus, right, as a shorthand, are transferred. Okay, so one atom gives, right, to another atom. So when you have a transfer of something with a negative charge, right, that negative charge leaves one atom and is added to another atom. So as an example, we have sodium, okay, and we have chlorine, and sodium is going to donate, right, or transfer an electron to chlorine. And so when that happens, that means sodium becomes positively charged because it loses a negative, right? Sodium becomes positive. And chlorine becomes what we call chloride. It becomes negatively charged. So we have a positively charged cation and a negatively charged anion, and it's the attraction of those 2 charges, right? So you don't really write them sodium plus and chloride minus. It would just be written NaCl. But it's the attraction of those oppositely charged ions that is what makes the bond, okay? Now, just review, right? What is the important particle for bonding? Right? It's always electrons. Whether we're talking about covalent bonding or ionic bonding, it's going to be electrons. Now, where do I find this number on the periodic table? So we didn't talk about that yet, right? So for an electrically neutral atom, meaning an atom of an element, the number of electrons is going to be equal to the number of protons. So when I look at the atomic number, not only does that tell me the number of protons, if it's electrically neutral, that tells me how many electrons also. So now we have a little illustration to show us the electrons in their shells around sodium and around chlorine and the transfer. Okay, so if we look at sodium, okay, we have 2 electrons in that first energy shell. We have 1, 2, 3, 4, 5, 6, 7, 8, 8 electrons in the second shell. So the first shell is full with 2, so it's full. The second shell is full with 8, right? So it's full. But then we have this one other electron, okay? So the octet rule says atoms are going to want to have their valence shell filled. Well, sodium, to fill the valence shell, it would either have to gain 7 electrons, right, to fill that outer shell, or it can lose 1. Much easier just to lose 1, right? We have a term, electronegativity, okay, which is a long word that means how much force or pull an atom has for electrons. So the higher the electronegativity, that higher that number or that value means the more tightly they hold the electrons. The lower that electron, the more likely they are. So sodium is going to have a low electronegativity value for this electron, right? Because it's going to want to give it up. Chlorine, on the other hand, is going to have a high electronegativity value. That's because if we look, okay, we have 1, 2 in the first shell, we have 8 in the second shell. In the third shell, we have 1, 2, 3, 4, 5, 6, 7. So there's just this one missing electron, and it would have a full valence shell. So chlorine is happy to accept that electron from sodium, and now we have sodium with 1 less electron. So we have a sodium ion, and we have chloride ion because we've gained a negative charge. It's the attraction of those that's going to make the ionic bond. Now, a covalent bond happens when atoms share electrons. Okay. Now, have these same questions. What's the important particle for bonding? Always going to be electrons. Where do I find that? Now we know we find it in the periodic table. Now, the interesting thing, though, that I want to make sure we don't forget about is even though atoms are sharing electrons, they don't always share equally, right? One atom may have more attraction and they may keep the electron around that atom more of the time, but it's not a transfer, right? There's no positively charged ions and negatively charged ions. It's just an unequal sharing. Now, do you remember, okay, covalent bonds, if we have unequal sharing like H2O versus equal sharing like O2, so oxygen with another oxygen. Do you remember what that's called? So pause your video and name that. So the unequal sharing is called polar covalent bonding, right? Polar meaning that they're not the same, right? Equal sharing would be non-polar covalent bonding, okay? Now we talked about water as an example of a polar covalent bond. So in this image, what we see is we see 3 different water molecules, okay? Now, let me just change colors. This, right, would be 1 water molecule. We have a second water molecule, and we have a third water molecule. Now, the covalent bonds are between the oxygen and the 2 hydrogens, right, for each water molecule. So these are the covalent bonds between, there's an oxygen, and each oxygen is bonded to 2 hydrogens for each water molecule. That covalent bond is unequally, the electrons are unequally shared, meaning the electrons spend more time closer to oxygen. So if I were to draw those electrons in, I would draw it a little bit closer to oxygen, right? They're still shared. Oxygen is still sharing them, but they're not being shared equally. And so that is a polar covalent bond that's occurring. And what does that do? That makes the oxygen atom in that covalent bond slightly more negatively charged. Okay, it's not an ion, it's just slightly more negatively charged. It makes the hydrogens slightly more positively charged. Okay? For all the water molecules in solution. Now, what that does is it leads to another type of bonding called hydrogen bonding. Hydrogen bonding is not occurring by electrons being shared or transferred. It's actually occurring because it is occurring between a hydrogen from So bonded to another atom. So a hydrogen that's covalently bonded to another atom and an electronegative atom that is also already covalently bonded to something else. So oxygen classifies, it is a electronegative atom. So that means hydrogen bonding can occur between water molecules. Okay, now I'm going to erase this because I have a bunch of stuff on here and I just want to clean the slate a little bit. All right, now we can look at it. All right, what that means is these little dashed lines, okay, those are the hydrogen bonds. So they're occurring between a hydrogen that's already bonded to another water molecule and an oxygen that is bonded to another water molecule. It's the attraction, right? Because we have a partially negative oxygen and a partially positive hydrogen. So it's that electrostatic interaction that causes those to form what we call hydrogen bonds. Hydrogen bonds are weaker bonds than a covalent bond or an ionic bond, but they are very important in living things, okay? So hydrogen bonds are made and broken all the time. Remember when we taught in your lesson, when you learned about hydrogen bonding, the surface area of water is in fact true because of hydrogen bonding. When we have adhesion and cohesion, that's because of hydrogen bonding. Its ability to dissolve and serve as a solvent is because of hydrogen bonding. The fact that ice, which is the solid form of water, spreads out, right, and is actually less dense than liquid water is because of hydrogen bonding, right? Hydrogen bonding is very important to living things. Now let's cover two terms. What does hydrophilic and hydrophobic mean? So hydro, I'm going to give you a hint, means water, right? So what do these terms mean? So stop your video there and just see if you can describe that and then come back. So philic, right, meaning loving. So philia, philic, loving. So hydrophilic would mean water loving. Hydrophobic, to fear of, right, water fearing. So that would mean it wouldn't mix well with water. So an example of something hydrophobic would be oil, right? Things that would dissolve in water, like polar compounds, ions, things like that would be hydrophilic. All right, we have now sort of gone through and covered some review for lesson 1 and for lesson 2. So if you haven't yet, this would be a great place to pause your video, take a break, do some jumping jacks, go for a walk, have a snack, and then come back and pick this up in just a little bit. Biological molecules, though, as we move forward, we're hitting on lesson 3. Biological molecules or macromolecules basically means these are the carbon-containing molecules in our cells that are carrying out all kinds of functions. They have monomers, right, which can be bonded together to form polymers. When we eat food, like this delicious pizza there, the food gets broken down into smaller particles. We absorb that, and then we use those smaller particles to build up larger ones. So let's get started into biological molecules. I first want to say, so we should definitely know the 4 classes, right? Carbohydrates, proteins, nucleic acids, and lipids or fats. Okay, so as I mentioned, when we think about biological molecules, we want to think about the fact that they have monomers or building blocks for each category or each type, and those can come together to form larger molecules or polymers. And they do that based on one of these reactions. So does the dehydration reaction build up monomers to polymers, or does the hydrolysis reaction build up monomers to polymers? Yeah, the dehydration reaction is going to build up the polymers. The hydration reaction or hydrolysis is going to break down the polymers into monomers. Now we use the term organic, right? What does that mean? It doesn't mean anything about no pesticides or something like that, like we think of in the grocery store. What does that mean? Pause your video. Yeah, it has to contain carbon and hydrogen. That is in fact what it means to be organic. So no matter how small or large a molecule is, if it has carbon and hydrogen, it's organic. If it doesn't contain those, it's inorganic or it's not organic. Now, I'm not going to go through all the details about biological molecules because if you want to review that, you can go back to lesson 3. But what I want us to see here is, remember I mentioned in the last slide, dehydration reactions or dehydration synthesis are how monomers are bonded together, right, to build up to be a polymer. These are generic monomers, right? These could be the monomers of a carbohydrate. These could be the monomers of a protein. But what happens is, through this process, a molecule of water is lost. That's why it's called dehydration. And a bond is formed between the two monomers. So we can see where that water, right, is coming from, and we have this new bond that's formed. The flip happens, right, when we go from polymer to the monomers, water's added in, and that bond is broken, and so we have two monomers that are released. Okay, so monomers are the building blocks, polymers when you put the blocks together to form different types of molecules. Now, I would like you to take a minute on your scratch paper and at least, so you might not need to have an example category, okay? But at least write the 4 biological molecules down, carbs, proteins, nucleic acids, and lipids, and see how many of the monomers you can name and how many of the polymers you can name. Okay, then we're going to go through them one by one, and then we'll give you another chance at the end to name them. We're going to start with carbohydrates. The monomer of a carbohydrate is called a monosaccharide, so mono, one sugar. Some examples that we talk about frequently are glucose and fructose. I have the structure of glucose here for you in 2 different forms. So there's the linear form, right, the chain, and the form that forms the ring, right? Many times in our illustrations, glucose will be drawn shorthand as just a hexagon or representing a sugar, right? That 6-membered hexagon might just be drawn to represent a sugar. Now, it is important that you know something about the polymers of carbohydrates. So I have 4 here that are important: starch, glycogen, chitin, and cellulose. What should you know about those? Well, first of all, they're going to be polymers, right? The actual structure of those polymers, which ones are branched or straight chain, that you don't need to know that. Knowing that they're polymers of monosaccharides, great, but not this specific structure. What I do want you to know is that 2 of these, starch and glycogen, serve as energy storage. So we eat a meal, right? We break that down, we absorb a lot of glucose. We need to keep our glucose levels in a normal range, right? That's part of homeostasis. So that glucose then needs to be stored away as glycogen in our liver and our muscle cells, right? So that's our energy storage. Then later, if I go several hours and I haven't eaten and I need some energy, that glycogen can be released and broken down into glucose. So that's an energy storage form for animals like us. Plants store their long-term energy storage as starch. That's why when we eat plants, right? We eat corn, we eat wheat, we eat something else, it's got a lot of starch in it because we're eating their energy storage. Chitin and cellulose are not energy storage. They are structural. So in other words, cellulose is part of what is found in a plant cell wall, makes up the structure. Chitin is structural in arthropods, so their exoskeleton. So this is a specific example of a dehydration synthesis for carbohydrates. So we see instead of it being the generic monomer that we saw a minute ago, it's 2 monosaccharides that are coming together to form a disaccharide, right? A molecule of water will be lost. So in the way it's illustrated here, here's our molecule of water, but we could have drawn it here, right? But we have a new bond between those 2 monomers, so we're building up the polymer. Here's these 4 polysaccharides. Again, you do not need to memorize the structure of these. Just remember which ones are energy storage, which ones are animals. So starch, right? What's that? Is it energy storage or structural? Yeah, it's energy. Plants or animals? Plants. Glycogen, energy or structural? Yeah, energy for animals. Okay. Cellulose, structural for plants. Chitin, structural for arthropods and for fungi, but mostly we just need to know structural. All right, so hopefully now you can fill in the carbohydrates, right? What's the monomer? It's a monosaccharide. Examples, glucose, fructose. What's the polymer? Well, we have 4, right? Starch, glycogen, chitin, and cellulose. Okay. The generic polymer would be called a polysaccharide. Now we'll move to proteins. All right, the monomer of a protein is an amino acid. There are 20 of these, okay? And this is the generic structure of an amino acid. I do want you to be able to recognize that structure. How will I recognize it? Well, I'm going to look for this central carbon that everything else is bonded off of. I'm going to look for an amino group. That's a functional group that we learned about, which is a nitrogen with 2 hydrogens. And a carboxyl group, which is a carbon double bonded to an oxygen with another OH. This R group right here is the part that can be different. Each of the 20, right, is a little bit different for this part. But all of this is the same for all 20 amino acids. All right. Then these amino acids come together to form the polymer called a polypeptide, which is also can be a protein. Okay, so some proteins are a single polypeptide. Some proteins might have more than one that come together. We have so many different types of proteins in our body. We have structural proteins. We have enzymes that are proteins that catalyze reactions in our bodies. Our hormones, also many of our hormones are proteins. All right. There is a special kind of bond. It is a covalent bond, right, where electrons are shared, but it's a special kind of covalent bond called a peptide bond that occurs between the amino acids. So when we're talking about a dehydration reaction, right? So here's amino acid 1, here's amino acid 2. When this molecule of water is lost, right, due to a dehydration reaction, and we have a new bond that's formed between those two, that bond has a very specific name called a peptide bond. Remember, we have polypeptides that we're creating, so we have the peptide bond that makes that connection. These are 4 example amino acids. Absolutely do not memorize anything about the R groups. I just wanted you to see some variation, right? They all have the amino group. They all have the carboxyl group. They all have the central carbon. They just vary based on the R group. Proteins have a very important structure. Their 3-dimensional structure is vital to their function. So when we talk about proteins, we talk about their level of protein structure. So we have primary, secondary, tertiary, and quaternary. Now, pause the video, see if you can write down really quick, primary, secondary, tertiary, quaternary, what does that mean as far as structure? Okay. The primary structure is just the order of the amino acids. So as that polymer is being built up, what order do they go in? Is it lysine and then glycine and then tryptophan? What order are they bonded? Okay. Where does that instruction come from? Okay. And we'll learn more about the details of this later in the semester, but there is a gene that instructs or codes for every protein. So the gene is saying this amino acid comes first, this amino acid comes next. Secondary structure is once you have all those amino acids bonded together, right? They are going to influence one another a little bit. So do they want to be next to each other? Do they want to be repelled? It's going to cause them to have what we call local folding, okay? So hydrogen bonding can occur. Are we going to get these pretty alpha helices or beta sheets? Like what do they look like next to each other? The tertiary is really the overall 3-dimensional shape, right? Once all of the local folding happens. Quaternary is not necessarily present in all proteins. If you have more than one polypeptide that is produced and then folds and then comes together, then you have quaternary structure. Not all proteins will have quaternary structure. Remember that I told you proteins, their 3-dimensional shape is really important to the way that they function. They can become what we call denatured, which means their shape is altered. So remember we said, what's the smallest unit of things that we consider alive? It's a cell, right? Well, a protein is smaller than a cell, right? It's a molecule. So a protein is not living, so it can't die, but it can become non-functional. And when they are non-functional, we call that denatured if its structure, its three-dimensional structure, is altered. Now, all proteins have an optimum condition in which they work best. So an optimum pH, in which they work best, an optimum temperature, an optimum salt concentration. And if the conditions get too far outside of that, then it's likely that those structures will become denatured. Now, sometimes people think, oh, well, all proteins are going to function best in pH 7. That's not true, right? Now, the proteins and the enzymes in our blood, yes, they're going around, 7.4 or something is going to be their ideal. And if it gets too low, right, pH 4 or 5, they're going to have trouble functioning, or too high, right, 8, 9. But think about in our GI tract, in our stomach. Our stomach has a pH of around 2. So enzymes that function there, right, they're going to have a different optimum pH. But regardless, when temperature, protein, pH, salt concentrations get too far outside of the optimum, then we may have denatured proteins and they no longer function. So we filled in the carbs. How about proteins? Can you do that one? So the monomer these are amino acids. You don't have to name examples, right? But if you want to, that's fine. Glycine, tryptophan, there's 20, right? You don't have to know those. The polymer would be a polypeptide, right? And those fold to become functional proteins. So we have so many examples in our body, but you don't need to be able to recall these, right? But collagen is a protein. We have amylase and other enzymes that help break down our food. We have hemoglobin that helps carry our oxygen. Okay, so lots and lots of examples, but you don't need to be able to recall those. Nucleic acids. This is our genetic material, right? The polymer would be DNA or RNA, and we'll learn a lot more. We have a whole lesson coming up about DNA and RNA. For now, what I need you to know is what's the monomer of a nucleic acid, and it is called a nucleotide. And this structure that I'm showing you on the screen is a single nucleotide. It has 3 components. It has a sugar, okay, and it'll be a little different if we're talking about DNA or RNA nucleotide, but it's a 5-membered ring, right? It has a phosphate group, so there's a phosphorus with 4 oxygens surrounding it. And it has a nitrogenous base, meaning this is a structure that contains nitrogen. You can see all these little nitrogens, okay? Do you need to memorize the structure of that? No, you do not. But no, the sugar, the phosphate, and the nitrogenous base are what make up this monomer. Now, I want you to recognize generally a nucleotide monomer, but you don't need to know all of the intricacies of where there's a carbon, where there's a nitrogen, where there's an oxygen, just that there's a sugar, a phosphate, and a base. So DNA is a 4-letter alphabet, ACGT. And RNA has also a 4-letter alphabet, but it has one difference. Instead of thymine, it has uracil. For DNA, we have 2 strands. And again, we're going to talk about this so much more later in this semester, but we have 2 strands that are held together by hydrogen bonding. They always pair in a certain way. A always pairs with T, and C always pairs with G. So more to come on nucleic acids. All right. Nucleic acids. Monomer is the nucleotide, right? And so we have A, C, G, T for DNA. We have U for RNA. Polymer would be our DNA, right? And our RNA, okay? Now our last category, which are lipids, there's more varied examples than what we saw in the others. So we're going to talk those through. I do want you to see that it does have a similar monomer and polymer in a triglyceride or a fat. So a triglyceride or a fat has monomers of 3 fatty acids and 1 molecule of glycerol. So those three fatty acids get bonded onto that glycerol, and that results in a fat or a triglyceride. Those can be either saturated or unsaturated. And on the next slide, I'll show you a little bit about what that means. All right, so lipids. What do we need to know about lipids? So first of all, these are all hydrophobic or water-fearing. They don't mix with water. So if you pour oil, cooking oil and water together, they're going to make two layers, right? They're not going to they're going to separate. They're not going to mix. Examples of lipids in living things are fats or triglycerides, which I just mentioned, oils, waxes, phospholipids, which we now know have something to do with a plasma membrane, right? And steroids. Do you need to know the structure of all these? Definitely you do not, okay? But I want you to see, when you see a whole bunch of carbons and hydrogens, so that's called hydrocarbons, that is going to be an indicator that it is hydrophobic, right? Not going to like water. Now, I mentioned to you that fats or triglycerides can be unsaturated or saturated based on the fatty acids. So up here is a saturated fatty acid, and here is an unsaturated fatty acid. So forget the length, how many carbons there are. What other difference do we see between those two? We see this double bond right here between two of the carbons. So if there is at least one double bond between carbons, we would call that an unsaturated fatty acid. So if we have more than one double bond, that would be a polyunsaturated fatty acid, right? So if you're looking at labels, that's what a polyunsaturated fatty acid means. Saturated means there's no double bond, so it is saturated with hydrogens, okay? So here is a little cartoon illustration of a fat or a triglyceride. I mentioned there is a molecule of glycerol and then 3 fatty acids that are bonded to glycerol. So these fatty acids could be saturated fatty acids, they could be unsaturated fatty acids. One thing that's interesting to make note that really applies to our life here is that 1 gram of fat, right, contains 9 calories, whereas 1 gram of a carbohydrate or a protein is going to have only 4 calories per gram. So if we're eating fat, we're getting more than twice the amount of energy per gram. But if we're storing away fat, then we're storing away more than twice amount, right, than what we would have to store as proteins or carbs. So fat is a good long-term energy storage. Phospholipids. Now, these are a little bit similar to a triglyceride or a fat in that they have these long fatty acids, right? However, instead of it being a glycerol that they're bonded to, they're bonded up here to a phosphate containing what we'll call a head, a polar head, okay? What this is it creates a molecule that has a hydrophilic component and a hydrophobic. So these tails are oil-like, right? They're hydrophobic. But this head, because it has this phosphate group, it's hydrophilic, so it's water-loving. So we have a molecule that has a water-loving side and a water-fearing side. These come together in layers, okay? So we have bilayers that make up our plasma membrane or our cellular membrane, where the one layer, the heads are going to point into the cell because the cell is mostly water, right? And on the other layer, the heads are going to point out to the external environment because the extracellular fluid is mostly water. Then sandwiched in the middle, we have this oily component, right? So we have the tails together that forms this oily layer. So we have this internal hydrophobic layer with hydrophilic layers on either side. And that's what makes up our plasma membrane of all living things, of cells of all living things. All right. Our steroids have these very similar 4 ring structures. All the steroids, so cholesterol is a steroid. It can actually be converted into testosterone and estrogen. These steroids are going to be signaling molecules. We recognize them as our sex hormones, okay? Now let's have a few wrap-ups here. So how do triglycerides or fats differ? So turn it off, answer that. So they can either be saturated, right? They can have saturated fatty acids with no double bonds, or they can have unsaturated fatty acids with double bonds. We just compared phospholipids and triglycerides, right? They both contain fats. However, the phospholipid has that phosphate group in the head, which makes it polar, so it makes it water-loving. All right, now we should know this. What type of bond, this should say what type of bond joins amino acid. Let's just say this. What type of bond joins amino acids? So that would be a peptide bond. Okay, a little more review here. So you pause it, see if you can answer these, and then come back. So a nucleotide, that's the monomer, right, of nucleic acids. And so this would be a sugar, right? It would be a phosphate group. And it would be a nitrogenous base. All right. What is the monomer of DNA? That's easy. It is a nucleotide, right? What is this functional group? So there's that phosphorus. This is a phosphate group. Okay, let's move on. All right, name this structure. Again, pause it, see if you can name it, and then come back. This is an amino acid. Hopefully you were able to recognize that, right? Here's my amino group, my carboxyl group, my central carbon, and there's that R. This is a steroid. I recognize it from those 4 rings. And this is a nucleotide. I can see my phosphate, my sugar, and my nitrogenous base. Now we're moving into lesson 4. So this might be another great place to pause, take a short break, maybe come back tomorrow. So cell theory, okay, this is pretty easy, but the cell is the basic unit of life. We've covered that already. Living things are composed of cells, and cells come from other cells, right? Pretty clear. Now, I do want to see if you can name, there are 4 components of all living cells. So whether we're saying a prokaryotic cell or a eukaryotic cell, they all have these 4 things. Can you name those? Okay. So they have a plasma membrane, right? Or a cell membrane that separates what's inside from what's outside. They all have to have DNA. They all have to have cytoplasm. And they all have to have these little machines called ribosomes that produce proteins. So those are the 4 components that all living cells have. All right, so we have checked this off the list. We know those. All right, now I do want to talk about this for a second. Cell membrane, plasma membrane, cell wall. Are those 3 the same thing? Pause it and come back. They are not. Cell membrane and plasma membrane are 2 terms that mean the same thing. All cells have that, right? It's made up of the phospholipids and it surrounds the cell. A cell wall is external to that. Not all cells are going to have a cell wall, right? Some will. It's more rigid, and it would be found outside of the plasma membrane. All right. Now, I'm not going to go through all of the organelles and their functions, right? So we have an animal cell, we have a plant cell. We have a lot of the same organelles found in those. These are both eukaryotic cells, right? So they have membrane-bound organelles. We have some organelles that are not found in both, right? So for example, the chloroplast. Animal cells don't have chloroplast, right? That's where photosynthesis occurs. Cell wall. Animal cells don't have a cell wall, okay? And we have this very large central vacuole in a plant cell that animal cells are not going to contain, all right? Now, we also have some components in an animal cell that plant cells are not going to have. So we have the lysosome that's going to digest old cell components and waste, things like that. We also have this centrosome, okay? And later in the semester, we're going to talk about cell division, and we'll see that that plays a really important role in cell division. Plant cells undergo cell division, but they don't have this centrosome component. So I'm going to stop our review there. Remember, look back at your study guides. Those are a great way to prepare. Look back at the quizzes that you've taken in each of the lessons. Review those, see what questions you missed, see what questions you got right. Make sure you're reviewing those. And as always, reach out with any questions, email, inbox, or come see me during office hours.